How to coat plates with PODS for PeptiGel experiments. PODS crystals are shipped sterile and dried in vials containing 50 million or 250 million PODS. Always handle PODS using sterile technique in a flow cabinet. PODS are typically reconstituted at 200 million PODS per milliliter in PBS and Vortex. First, aliquot the desired number of PODS for your experiment into a new microcentrifuge tube. Perform any dilutions required in PBS. Vortex PODS solution and dispense into wells of a multi-well plate. Spin the multi-well plate in a centrifuge with a plate rotor for 20 minutes at 3000 G. Remove and discard the supernatant from each of the wells. Leave the well plate with the lid off under sterile conditions for an hour to allow PODS to dry. Alternatively, leave in the fridge with the lid on overnight. Once dry, inspect the wells with PODS under a microscope. The PODS should be uniformly dispersed across the surface of the wells. Select which PeptiGel you would like to use for your experiment. Allow PeptiGel to warm up to room temperature. Using a positive displacement pipette, transfer PeptiGel to a new microcentrifuge tube. Add cell culture media either with or without cells, depending on your experimental design. Ensure your final mixture has a 1:4 media to gel ratio. Mix by filling the pipette with gel from the bottom of the microcentrifuge tube. Then slowly release near the top of the mixture. Always maintain the pipette tip within the mixture to minimize introducing air bubbles. To remove bubbles after mixing, centrifuge at 2500 G for 1 minute. Dispense PeptiGel and cell mixture into wells of a multi-well plate. Ensure the entire surface of the well is covered. Add enough cell culture media to each well to cover the PeptiGel. Place the well plate in the incubator at 37 degrees Celsius. Change the media 2 to 3 times within 1 hour to ensure homogeneous diffusion of media nutrients.